Good morning, children.
Good morning, ma'am.
Good morning,
Morning,
ma'am.
ma'am.
We completed the first division of plant kingdom, that is algae, that we have completed. Now we have to start with the second plant division. So before we proceed to the second division, we will listen to the seminar of some of your classmates. So Alentina can start. Alentina, are you ready?
Yes, ma'am.
Okay. You can begin. Your topic, Alentina?
And the introduction of biological classification.
Okay, and don't make your classmates sit idle. Ask them something in between, okay? Keep them engaged. Yes. Shabari, Sara.
Ma'am, is it see? Ma'am, is my PPT
I can.
seen?
Yes, dear, I can see.
Okay. Good morning to all. My name is Anjina and I'm here to take a seminar on the biological classification from the chapter biological classification. So first, let us know what is biological classification. Biological classification is the system. It's a scientific method of arrangement of organisms into groups. So first we'll see Aristotle's classification. So he was the first to try out this classification based on scientific basis. So he used symbol morphological characters. Symbol morphology means the structure, and he divided plants into herbs, shrubs, and trees. And in animals, he divided into two, that are with red blood and without red blood. So here with red blood also can be said as vertebrates, those with vertebrates, and those without blood can be also said invertebrate. So for examples of vertebrates are viviparous quadrupeds and oviparous quadrupeds, whales, fishes, etc. Whereas without red blood, they can be shelled animals, cnidarians, spiders, scorpions, etc. So here are few examples of red blood and without red blood. Here you can see amphibians, fishes, reptiles, birds, animals can all come under red blood. Whereas in invertebrates, here are few examples of flatworm, jellyfish, which is a cnidarian, spiders, snail, shrimps, etc. Then two kingdom classification. Two kingdom classification was proposed by Carolus Linnaeus in 1785, and this system classifies organisms into two kingdoms, which is kingdom Plantae and kingdom Animalia. And this had several drawbacks. The first drawback was that it did not distinguish between prokaryotes and eukaryotes. So prokaryotes are those which do not have well-defined nucleus. As you can see in the picture, the genetic material is in direct contact with the cytoplasm, whereas eukaryotes, the nucleus is enclosed within a membrane that is called nuclear membrane. And then it did not distinguish between unicellular and multicellular. Then earlier classification included bacteria, cyanobacteria, fungi, mosses, ferns, gymnosperms, and angiosperms under plants only on the presence of cell wall. And this again had a drawback. That was the cell wall of different organisms had different composition. For example, fungi had chitin in their cell wall, and the plant cell wall was made up of cellulose. Bacteria was made up of peptidoglycan, or for algae it was mannans, galectin, and many other minerals. And he did not differentiate between heterotrophic and autotrophic. Example for heterotrophic are fungi, whereas in autotrophic were green plants, and large number of organisms did not fall into either of these categories. Then five kingdom classification was proposed by R.H. Whittaker, and his main criteria was cell structure, thallus organization, mode of nutrition, whether they were heterotrophic or autotrophic, reproduction, were asexual or sexual, or phylogenetic relationships. And the five kingdoms were Kingdom Monera, Kingdom Protista, kingdom fungi, kingdom plantae, and kingdom animalia. And let's briefly have a look on these 5 kingdoms. So, kingdom Monera, they were unicellular and prokaryote. But it's actually not nucleus, it was nucleus membrane. Their nuclear membrane is absent because they are prokaryote. And the body organization is cellular level. Mode of nutrition is autotrophic or heterotrophic. An example for Monera is bacteria or blue-green algae. And for protistas, they are unicellular eukaryotes, and so do they have nuclear membrane present. And the body organization is cellular level. The mode of nutrition can be both heterotrophic and autotrophic. Example are Spirogyra and Chlamydomonas. Fungi, they are multicellular, non-green because there is absence of chlorophyll, and eukaryote. The nuclear membrane is present. They are multicellular with loose tissue, and the mode of nutrition is heterotrophic, and there's saprophytic, parasite, and some symbiotic. Symbiotics example can be lichens, which is in association with fungi and algae, and the examples are Rhizopus and Agaricus. Kingdom Plantae, they are multicellular and eukaryote, and they have nuclear membrane present, and they are tissue level and organ level, and they are autotrophic, and examples are herbs, shrubs, and trees. And Kingdom Animalia, multicellular eukaryote, their nuclear membrane is present, they are organ level system, and their mode of nutrition is heterotrophic, and examples are fish, frog, crocodiles, human beings, etc. Thank you.
Any doubts, children? Any doubts? No doubts. Mutual understanding, no? You will not, we will not ask you any doubts and you also should not ask. Is that the reason? No doubts. Sure? Why some are smiling? Such an understanding was there among you people? Maybe I don't know. Okay. And Alentina, well done. And I just want to say one thing. Alentina, you have done well, but I felt you also went a bit fast, just like our Nimida. You don't have to be in a hurry. You can be very relaxed and you can take and you can include participation from your classmates also. So next time when you're preparing the seminar, see that somehow you are involving their participation. Okay? Agreed? All have done so far, all have taken their seminar well, and I will give chance to one more person, and the third one can take in the last, second last period. Who is going to take next? Gaya or Samia?
Ma'am, I will take next period. I won't be there.
Okay, then you can. But Samia, your video is not there, no? Please turn your video on.
Yes, ma'am, I will.
You may start. What is your topic?
I'm Kingdom Monera.
Okay. You don't have to be in a hurry. Take it peacefully. Nimida, Shabari, Anashwara, Anne Mary, Abhinav, Sreya, Agna. Samia, unmute.
Good morning. I'm going to take seminar on Kingdom Monera. So before I start, I would like to give a small idea about this. Throughout this, we'll be going to see about bacteria as they are the main members or the only members maybe in this kingdom. So throughout, we'll be seeing this. Seeing about bacteria. So as I have told, the kingdom consists of mainly. So as I have told, this kingdom consists mainly of bacteria, and they are the most abundant microorganisms compared to the other microorganisms. And their presence can be found everywhere, including all the extreme and harsh habitats, like from icebergs to the volcanic lava. Everywhere they can be found. Then many of them are parasite. Majority of them are parasites. Yeah, so this picture is of bacterial cell anatomy, and As we can see, compared to any other organism, the structure is very simple. Like when you compare it with any other multicellular or even unicellular organisms, the structure of bacteria is very simple, but they are complex in their behavior, and they have an extensive metabolic diversity compared to many other organisms. Like with other organisms like human beings, not even human beings, like multicellular organisms like ants and all, they have a very small, they have a very simple structure, but they have an extensive metabolic diversity. And they have been older than, the oldest non-life form on Earth. Like they have been more than, they have been on Earth for more than 3.5 billion years. And this makes them the oldest non-life form on Earth. Yeah, so now we move on to the classification of bacteria on basis of different things. First, we'll see the classification of bacteria based on their shape, then on the basis of their food intake, and like one more classification, which is the main classification of bacteria. So first, on the basis of their shape. So there are four different shapes of bacteria, like cocci, bacilli, spirilla, and vibrio. Cocci is sphere-shaped or like ball-shaped. Then bacilli is rod-shaped, and spiral, as the name suggests, is it's spiral shape. Then vibrio is in the shape of the punctuation mark, comma, and one second. Yeah, so here you can see the singular names of these, like cocci becomes coccus, bacilli, bacillus, vibrio, vibrium, and spirilla, spirillum. Yeah, so here first two names you replace the I, that is cocci and bacilli, you replace the I and add US to get the singular name. Then in the second two cases, first case you remove IO, and the spirilla you remove A and add UM. So you get their singular forms, like cocci becomes coccus, bacilli becomes bacillus, and vibrio becomes vibrium, and spirilla becomes spirilla. Then we move on to the class. Sorry. Before that, many of the food items which we eat are made up of bacteria. Then classification on the basis of food intake. So as we know, there are two main modes of nutrition, that's autotrophic mode and heterotrophic mode, and both these can be found in bacteria. Then when you come to autotrophic, both chemosynthetic and photosynthetic can be found. Chemosynthetic means they use organic matter, and photosynthetic they use organic matter, and chemosynthetic they use inorganic matters. Like chemicals, and in photosynthetic they use water and oxygen. Then heterotrophic mode of nutrition, it's parasitic and saprophytic. Parasitic, they take nutrition from a living organism, and in saprophytic, they take nutrition from a dead and decaying matter. Now we come into the main classification of bacteria, which is like when the three domain classification came, bacteria was split into two different domains or two different kingdoms again. And so bacteria can be divided into archaebacteria and eubacteria. Now bacteria is fast there because they can move 100 times its own body length in a second. Whereas when you take a big fish like shark or dolphin, it can move only 10 times of its body length in a second. Then if you take all the bacteria in the earth and you join it end to end to form a light, it would stretch up to 10 billion light years. Now we'll see about archaebacteria. Yeah, so they are unique because they live in the most harsh habitats. So in the starting, we have told bacteria can be found in all the extreme habitats. And these bacteria which can be found in these harsh habitats are archaebacteria. Like for example, in extreme salty areas, halophiles are found, and hot springs and acidic areas also thermoestophiles are found, and marshy areas methanogens are found. And the methanogens are found in the gut of several ruminant animals. Ruminant animals, they just digest their food twice, like they have a two-chambered stomach because they have to um, digest cellulose, which is found in grass. So ruminant animals are mainly the grass-eating animals, and methanogens can be found in the guts of ruminant animals, and they are responsible for the production of methane or biogas from the dung of these animals. Then they differ from other bacteria in having a different type of cell wall, and this unique cell wall helps them to exist in helps in their existence in these harsh habitats. Now here you can see the pictures of the three types, like halophiles, thermoestophiles, and methanogens. First one is just not a microscopic picture. The second two are microscopic ones. Then we move on to eubacteria. Yeah, eubacteria is also called true bacteria, and they are characterized by presence of the rigid cell wall. And if they are capable of movement, flagella can also be found. Yeah, now we see different types of eubacteria, like all three types of nutrition can be found in, we told, in bacteria. Like we are going to see different bacteria with different modes of nutrition. First one, we'll see photosynthetic bacteria, which comes under eubacteria. Yeah, cyanobacteria is also called the blue green algae, and what they are photosynthetic because they have the presence of chlorophyll A, which is similar to that of plants, and they are unicellular, and they can be found as colonial forms or filamentous forms. And if colonial, they are surrounded by a gelatinous sheet, and they can be found in freshwater, marine water, and terrestrial areas, and they form algal blooms in polluted water. This is a picture of an algal bloom, and this can affect the marine life by making the availability of oxygen very low. And some of these fix atmospheric nitrogen in specialized cells called heterocysts. For example, Anabina and Nostoc are two types of cyanobacteria. This is a picture of Anabina, and this is a picture of Nostoc. Yeah, now we come into chemosynthetic or bacteria, which is coming under eubacteria. And this bacteria helps oxidize various inorganic substances. So we know chemosynthetic, they use chemicals. So that is why they use, they oxidize various inorganic substances, nitrates, ammonia, and use the released energy for their ATP production. And they play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Then a human body consists of more bacterial cells than human cells. They help with processes such as digestion, and they also defend our body from other bad bacteria. And out of all the bacteria present on Earth, less than 1% will make us sick. Yeah, now we move on to heterotrophic bacteria. They are the most abundant in nature because, like we told in the starting, parasites are the majority of bacteria, and heterotrophic bacteria, they are the most abundant, and the majority are important decomposers. Many of them have a significant role in human matters, and they are helpful in making curd, then the production of antibiotics, and fixing nitrogen in legume roots. Like production of antibiotics, like bacitracin, and bacitracin is made from a bacteria called Bacillus subtilis, and an interesting thing about this is that production of antibiotics is, of course, antibiotics is used against bacteria, and they are made from bacteria itself. And some are pathogens which cause damage to human bodies, and farm animals, crops, and all. Then bacteria was discovered in 1674 when Dutch scientist Antony van Leeuwenhoek quoted them as he was looking at scrapings from human mouth under a newly invented microscope. Now we'll see about mycoplasma. Mycoplasma, they completely lack a cell wall, and they're smallest living cells ever known, and they can also survive without oxygen. Many of the mycoplasma are pathogenic. Then bacteria adapts quickly to any environment, and like if you are using a particular antibiotic for a longer time, that bacteria will get resistant to it and it will be no longer useful. Now, reproduction in bacteria. Bacteria reproduces mainly by fission, and this is just a representation of fission. And under favorable conditions, they also reproduce. Under unfavorable conditions, they reproduce by producing spores, and they also reproduce by a sort of sexual reproduction by adapting a primitive method of DNA transfer, like from one bacterium to another, the DNA, the genetic material is transferred by conjugation, and this is with the help of a structure called phyllis. I don't know whether you have noted that, because we have gone through the picture of bacteria now. Yeah, here you can see. Here you can see phyllis, and that helps in the reproduction also. The sexual reproduction in sexual reproduction in bacteria. That's it from me. Thank you.
Samia, very colorful presentation. I think you have taken so much effort in preparing the presentation, and the way you added all those extra points, the curious facts, that also I like. So it was good. What about Gaya? Are you ready? Gaya, I think you can also complete it, because hardly 15 minutes are present. Sorry, 15
Ma'am,
we have. Yes.
ma'am, I'm also assigned with a seminar.
Okay. I think we'll have time. We have 15 minutes more. Who wants to take first, Sara or Gaya? Who is more kind-hearted? Okay, Gaya, you may take. Your topic?
Viruses, viroids, and lichens.
And by the way, anybody helped Samia to prepare, or you did yourself? Samia? Huh?
I did myself.
Good. Yes, continue. Children all should turn your videos on. Just 1 minute.
Ma'am, can I start?
Yes.
Okay, good morning to all. Today I'll be taking the seminar on the topic viruses, viroids, and lichens. Okay, so we learned about 5 different kingdoms, right? Okay, so there are certain other organisms that cannot be placed under any of these kingdoms, and they are viruses, viroids, and lichens. Okay, so they are not included under the 5 kingdom classification, and the reason is because they are acellular organisms. They are non-cellular. They don't have a proper cellular structure, and they don't have a cytoplasm. They don't have a cell wall, and so we cannot consider them as living organisms. And they have an inert crystalline structure outside the cell. They don't react when they are outside the cell. They only react when they enter into the body of a living organism. And when they enter into the body, they will start reacting. They will start multiplying. Then they will cause diseases, and sometimes they can also lead to the death of that organism. And they are obligate parasites. They depend on other organisms, and they lead a parasitic life. And okay, so first we have DJ Vanowski. He was the first person who discovered virus. and he coined the term virus, which means venom or poisonous fluid. And and he also recognized that there were certain microbes that were causing this tobacco mosaic virus, and so he wanted to find out the organisms that were causing this. And so he created an extract out of this infected plant, and he just he created an extract out of the infected plant, and he passed the solution through bacteria proof filter so that he could separate it and find out what the organism was. But he couldn't find the organisms. He couldn't find the organisms, and it was because they could pass through bacteria proof filters. So he concluded that they were smaller than bacteria, and because they were able to pass through bacteria proof filters. And then we have M.W. Bejanic. He demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants. Okay, so he took the extract. He made an extract out of the infected plant of tobacco, and then he used it. It was like a fluid, and then he used it. He used it on a normal healthy plant, and he found out that this healthy plant also started to show the symptoms. The healthy plant also uh, started to show the symptoms, and he understood that it was spreading. So he understood that it was contagious. So he called it contagium vivum fluidum, which means infectious living fluid. And then we have W.M. Stanley. He showed that the viruses could be crystallized, and the crystals were largely made up of proteins. So now the structure of the virus. A virus is basically a nucleoprotein, and it is made up of nucleic acid and a protein coat. And this nucleic acid can either be DNA or RNA, and there is also a protein coat called capsid, which protects this nucleic acid, and it is made up of small units called capsomeres, and they are arranged in helical or polyhedral geometric forms. um, the virus is a nucleoprotein, and the genetic material is infectious. It can cause diseases. Okay. Like I said, the genetic material can either be DNA or RNA. So the viruses that infect plants will have single-stranded RNA, while the viruses that infect animals can have either single-stranded or double-stranded RNA or double-stranded DNA. And the bacteriophages, they are the viruses that infect bacteria. They will have double-stranded DNA. Okay. Now we have viroids. Viroids were discovered by Theo Dinner in 1971, and they were infectious agents. And the difference between viruses and viroids is that viroids will be smaller than viruses, and they don't have any cell wall or protein coat or anything. They just consist of RNA. The genetic material is RNA, and it is of very low molecular weight. And and they are infectious agents. They can cause diseases, and one example is potato spindle tuber disease. Here, the shape of the potato will change, and it will become like a spindle shaped. Okay, and yeah. Next, we have lichens. Lichens are the symbiotic association between algae and fungi. So, symbiotic association can be like two organisms will be involved, and it can be like both the organisms are benefited, or where one organism is benefited and the other is not. So, it can here both organisms are benefited, both the organisms are mutually benefited, and we have two organisms, algae and fungi. And the algal component is called phycobion, and it is autotrophic, so it is capable of preparing its own food. And the fungal component is called mycobion. It is heterotrophic, it cannot prepare its own food. And so here the algae will perform the photosynthesis, and it will synthesize the food and give it to the fungi, and the fungi will in return it will provide shelter, and it will absorb the nutrients and water and all, and it will provide it to the algae. And they are usually seen growing on trees and on rocks, and they are not found in polluted areas. They are usually seen in pollution free areas, in areas of less pollution, and so they are considered as good pollution indicators. Okay, so that's it. Thank you.
Here also no doubts, no? You don't have any doubts. Okay, Gaya, it was a good presentation. Now, who is next? Sarah? I think you will take how many minutes, Sarah?
Ma'am, it will be, I think, 7.
7 minutes. So we'll be able to manage. 5 more minutes we have.
Yeah.
Ah, okay. Then you can start. A few of you haven't turned your videos on. I'm noting all these names. Oh, same topic.
Yeah. My goodness. I'm keeping. Is my voice clear, ma'am? Ma'am?
Yes, yes, I can.
Okay. Good morning to all. Today I'm going to take, today's topic is virus, viroids, prions, and lichens. So let's start. The, Before that, let's recap that these are the 4 words that it's not there in the five kingdom classification of Whittaker, because some of them are acellular, some of them are non-cellular, and which made them does not fit in neither of the kingdom. So for a recap, let's find the difference between virus and bacteria. So viruses are very small, non-cellular, have no metabolism, neither grow or divide. Bacteria can be crystallized, command the host cell to produce virus, all produce disease in man, animal, and plant, man means human. In bacteria, they are large, can be seen by a light microscope, single-celled, have metabolism, grow in size and divide to produce more, cannot be crystallized, self-produce, some are harmless, some are useful, some are disease-producing. I hope you understood the difference between virus and bacteria. Okay, so let's start with virus itself. Viruses are non-cellular, obligated parasites that are characterized by having an inert crystallized structure outside the living cell. Non-cellular, they don't produce their own energy. Obligated parasite, they cannot survive without they don't survive alone, independently. That are characterized by having inert crystallized structure. Once a virus infects a cell, they kill the host cells and take over the machinery, means human or any host of the host cell to replicate themselves. Mm, about this, if you guys read newspaper that was updated in COVID-19 days, like 2019, it will be much clear. The name virus meaning venom or poisonous fluid given by Pastor DJ Ivansky. Yeah, he discovered it. And the meaning of it is venom or poison. DJ Ivansky recognized certain microbes as a causal organism of methanic disease of tobacco, where he found to be smaller, where it's found to be smaller than bacteria because they pass through a bacteria-proof filter. W. Benjanik demonstrated that the extract of infected plant of a tobacco cause infection in healthy plants and called the fluid as containing viva and fluidum. The last 2 points, I'll say in simple words. The virus was invented by Ivansky in 1892, and later it was proved by W. Benjanik. This is how he did that. That's it. Okay. Same topic. MS Stanley showed that the virus could be crystallized, and crystallized consists of largely proteins, which are inert outside their specific host cell. They have crystallized. He proved that they have crystallized, and crystal consists of large proteins. In addition to proteins, virus also contain genetic materials that could be neither RNA or DNA. Yeah, they also contain genetical materials. And why there is a or between RNA and DNA, I'll say. Virus that infect plants have single-structured RNA. That's why there's a or. Virus that infect animals have neither single or double-structured RNA or double-structured DNA. Bacteriophages are usually double-structured DNA. A fun fact: their meaning of bacteriophages are bacteria eater. Yeah, bacteria eater. The protein core the protein core is called capsid, made of small subunits called capsomers, protects the nucleic acid. A virus cause diseases. Let's take a simple one: flu, that we all have like been through once in our life, and smallpox, herpes, influenza, AIDS. Different shapes of viruses and the structural components of viruses. These are two pictures. Go through it. I hope you're done. Okay, what are viruses and lichens? Viruses are smaller than viruses, which are found to be free in RNA, and viruses lack in the protein core that is found in virus, hence they are named viroids. One of the disease caused by viroids are potato spindle tuber disease. And what are lichens? They are a symbiotic association between algae and fungi. Symbiotic meaning interaction between two organisms that are physically close. Algae compound is known as mycobiotin, and fungal compound is known as myco. Oh, phycobiotin and fungal compound is known as mycobiotin. Algae prepare food for fungi, and fungi provide shelter and absorbs minerals, nutrients, and water for its partner. Lichens are very good pollution indicator. Pictures of lichens. This is what they look in real life, and this is what they look in textbook. Prions. The word prions derives from a proteinaceous infectious particle. It's not mentioned in the textbook, it's just extra. Prions are infection agent responsible for several neurological diseases, were found to be transmitted by a consisting of abnormal folded proteins. The agent is similar in size to virus, that's why they are known as prions. They do not have a nucleic acid. Disease caused by prions are scrapie in sheep, mad cow disease in cows. The scientific name for mad cow is bovine spongiform encephalopathy, and a chronic wasting disease in deer. An analogic variant of mad cow disease caused in humans are cruciferal Jacobin disease. This is scrapie found in sheep, and this is cruciferal Jacobin disease found in human. Thank you.
Okay, Sarah, a little extra time, no problem. You were very cool and relaxed, but at times I was not able to follow some of the, like the pronunciation of certain, maybe it is because of the audio problem, I don't know. I was not able to understand certain terms. Other than that, it was good. And where that red mustache is gone now, throughout the session, it was there. It's your headset.
Yeah, ma'am.
Okay. Okay, so only that. All have completed, no? All have completed the seminar. Those who were assigned, I think, they had completed. So see you next period. So next period, we'll continue with the chapter Plant Kingdom. Okay, thank you, children. See you next class.
Thank you.